In this video, we will discuss about this CRISPR genes, identifies genes that are important in this 3D migration. So, in the previous video, we have discussed the genes important in the migrations and proliferations in 2D model. Here, they did the same study, but in 3D amyloid model, and they do the same analysis. That is, they check the proteins that or genes that are differentially regulated in the chemokinesis and the amyloid. And what they are able to find out is that the genes that are differentially regulated in the 3D model is different from the 2D model. And one of the reasons could be because in 2D models they have these integrins that can interact with the cell surface or surface in the on the 2D models, whereas in the 3D models they don't have such integrins interactions to the surface. And however, the genes, they check some of the genes that are differential or some of the gene knockouts that shows the highest migrations. So out of this they selected two different genes, that is COROA1 and fNL1, and also this TLN1 and ITBG2, they are not that much differentially regulated. The role of TLN1 is to provide interactions between the cytoskeleton and the actins that are present in cytosols. So disruptions of this will affect the migrations in the 2D models. So when they checked the knockdown of this TLN1 and generated stable cell lines and checked its checks the impact of those stable cell lines in the 3D model, they are not able to see any significant changes. And this ITBG2 is used as a control. So first, what I check is the median speed. And what they did is they developed the stable cell lines for all these four knockout that they have identifies in this chemokinesis in the amyloid 3D model structure. And when they check the and they again run this stable cell cell lines in this 3D amyloid model, and then they measure this median speed of the cells migrations, and what they're able to find out compared to the control, that is compared to the control, and that does not have any knockout, and the ITBG2 knockout, the median speed is very low in the or significantly lower in the case of this CORO1A knockout and fNL1 knockout. It can be seen in this panel B left hand side. And when they check the persistence, it is directional mobility in the same directional movement, or in other terms movement in the particular directions. What they are able to find out for this COROA1, the direction persistence is high, whereas this fNL1 the persistence are lower. So from this it seems like that fNL1 does not have that much role towards fNL1 has a role, we can say, towards maintaining the persistence, and its knockdown will affect its persistence, that is a directional movability. And for the CORO1A, we can see even if the CORO1 knockdown is there, do not see too much impact in the cell's persistence. Next, they wanted to check its locations within the cells during these migrations. So when they check it for fNL1, they see that fNL1 are mainly lies in the backside or in the back of the cells during the movement. It can be seen here and in the panel C. So this indicates that this fNL1 might help in the this morphology change of this of the cells during the persistence or migrations. And for the CORO1A, they are able to see its presence both in the front end and also in the end back end, and this code indicates its role, this CORO1A role Also towards the cells enlargement or cell shrinkage that is important during the migrations of the cells. So this is what these two genes they have find out that plays a role towards the cells migrations in the 3D model. I hope you understood how they come up to how they check these gene knockouts roles and how they come out to these gene knockouts using the 3D amyloid model studies and comparing the chemokinesis in the amyloid 3D model. If you need to understand more about their experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.